I'll introduce you Dr. Takis Benos. Dr. Benos is the William Bushnell Presidential Chaired Professor in the Department of Epidemiology at the University of Florida. He received his Ph.D. from the University of Crete, after which he was a postdoctoral fellow at EMBL with Michael Ashburner. He then came to Washington University in St. Louis to work with Gary Stormo to develop computational methods to model protein-DNA interactions. Dr. Benos joined the faculty in the Department of Computational and System Biology at the University of Pittsburgh, where he remained until 2022. His lab focuses on developing computational methods for systems medicine, particularly tools to integrate multi-omics and clinical data in order to understand the biological processes underlying disease and aging. So now let's welcome Dr. Benos.
Thanks, everybody. Can you hear me okay? Yep. Okay, great. So thanks for the introduction. Thanks for inviting me. Thanks for organizing the whole thing. I'm going to talk to you today, not so much about the research we do. Maybe I might touch a little bit on that, but I'm going to talk mostly about my experience with Gary. So this, for those that wondering, yes, this is a picture of Gary. And when he was a bit younger, and in a rock band, hence the title. And I will, if we have time, I'll tell you a little bit of a story how this band changed his life. So I met Gary while I was at EMBL EBI doing a postdoc with Michael Ashburner, who was a very well-known Drosophilist, but also pioneered the gene ontologies, open access journals, and that stuff. And at that time, I think it was about 1998, we had there was a symposium that Gary participated in the Isaac Newton Institute for Mathematical Sciences. And I remember two people that participated, Gary and Anders. Gary, because he took us out for lunch, all the younglings that were there, the students and the postdocs. And I was surprised because he not only was able to very articulately express his research, but also listen to the nonsense that we were throwing back to him in form of questions or suggestions even, and patiently thinking about them or pretending to think, and then responding to us in a very kind way. The reason I remember Anders is because he was talking about Markov models at the time, you know, the hidden type, the shy ones. But I remember Anders not so much about the forward-backward algorithms, which always confused me. I thought always the algorithms are forward thinking, but he surprised me because at the end, he thanked Nokia for funding his research. And when I asked him what happens, why in a bioinformatics symposium you thank Nokia, said, well, Nokia, the data that I received from Nokia to develop the hidden Markov models are much more rich and much better than the biological sequences, which were very limited, as we know so far. So, and I forgot to say, so then I was very much impressed with the work that Gary was doing. So I said, I would like to apply to go to his lab. So when I went back, I wrote a very nice letter saying I was very much impressed with the work that you do. This is something I wanted to do. So would you accept me in your group? I also knew that he was in Boulder, Colorado, which was a very nice place, you know, a lot of mountains, you can do skiing, you can do a lot of things. So I was really very excited. So sure enough, 1 week after that, no response. 2 weeks after that, no response. I think it took him about 2 months or 2.5 months to send me a response of that. And when I saw, you know, response from Gary Stormo, I was very excited. Response from Gary Stormo, response from Gary Stormo. I opened the email and says, sorry, it took me long to respond. Yes, I have an opening in my lab and I would like you to come. Even more excited. But I'm moving my lab from Boulder to St. Louis. Not so excited. Not so excited because, you know, St. Louis, I didn't know much about St. Louis, but there was not too many mountains around here, are there? So anyway, so I came here. Because, yeah, of course, Gary, but also St. Louis was, as we heard from Son in the morning, there was a collection of very good people here, Bob and Son and Warren and Michael Zucker and David States and others. I thought it would be a very nice place, nice experience. And I started working with Gary, and then in the first couple of months, I received an email from Celera, which was a company at the time founded by Craig Venter, who threatened to sequence the whole human genome in like 5 years or so. And as a proof of principle, he said, I will sequence the Drosophila genome in 5 months. So sure enough, they have done that. And they asked me to go there because of my previous experience with the annotation of the Drosophila at the EMBL EBI. And Celera was the company that had the logo that, or the philosophy, shotgun first, ask questions later. It was the first genome that was done, the first multicellular complex genome that was done with shotgun sequencing. Nobody believed that this will happen. So I went to Gary's office and I say, I received this invitation from Celera, can I go? And this was only 1 month or 2 after I arrived in his lab, right? So he looks at me very puzzled because I didn't make clear that it's only for 1 week I want to go. So he looks at me and says, you just came here. What do you say? So anyway, but the good thing, one thing I learned from that thing, it was that he not only he didn't object to go, but he didn't criticize, didn't give me any grief or any hit about there, because there was a big fighting then between the public project, which was headed, among other people, from Bob Waterston here, and the Celera project. Unlike other people in the department who gave me a little bit of heat, I'm not going to mention names. I promised Warren that I'm not going to mention him in the talk. So I went to the Celera. We published. I'm part of this somewhere lost there on that. It was nice. And then I continue working on this famous protein with the paper that you have seen today few times, I think, and Martha and Scott and Zipeng that had, was to look on these zinc finger proteins. So do we have anything here to point? No. Okay, not that one. Okay, that one. So oops, how do you go back? It's a little bit delayed. Okay, so we're looking at these zinc finger proteins, and the idea is that we had some data from the wild type and some mutants with their DNA binding preferences. We feed them in this kind of Boltzmann distribution, and we published this paper. This paper, the interesting thing that it has is that Gary made very clear that what we had there on the Boltzmann distribution has an interesting implication when you use the PSSM models or PWM models, which is when we sum the scores of the PWMs, essentially, we add the potential energies on these sites. That's because the scores is the log probability, which will be the negative log probability, which will be the energy, right? So the additivity assumption was kind of very strong for us. And that's so we were surprised when we saw Martha's paper that she talked in the morning that said that additivity is not valid. And then we asked for the data, and I looked at the data, and I noticed that it was not exactly like that. So Martha had done the wild type protein and 4 mutants. And as you can see, the predicted probability with the measured probability, they do match fairly well. With the highest probability, you lose a little bit from the diagonal, but for the most part, you are doing pretty well. It's this protein that the additivity does not hold. And this protein had a non-specific binding. So when I saw it, I was a young to-be scientist. I was very combative scientist. So I said, okay, let's write a paper to tell them that they are wrong. And Gary said, no, let's talk with Martha and let's try to figure out what's going on and what happened there. And that's how this paper came into existence. There you go, which we saw that additivity is kind of good approximation, as Martha said in the morning. And also when we were doing this paper, we realized that in order to compare the models, we needed to develop a new statistic for comparing the information content of the models with different parameters, because the mononucleotide with the dinucleotide and so on, they had different parameters, obviously. And so Gary developed a method. Noticed that some of the statistics that we use follows the chi distribution. So we had a method to compare the models, and we proved that the additivity is a good assumption. Correct me, Gary, if I'm wrong at any time. Yeah. Don't be afraid that you will disrupt my train of thought. I don't have any. So then we moved to, I moved to Pittsburgh. I graduated from his lab. So I moved to Pittsburgh in 2002. And I continue, and I started applying for grants, right? I applied to NSF grants, right? We all need grants. I applied to NSF in order to extend this model that we have developed for the EGR proteins to other protein families. And some of the reviewers push back and say, no, it cannot be done on other protein families. So when we resubmit, Gary wrote this very nice letter, which among other things says, nobody knows whether such a model can be applied to other protein families. No one will know unless the effort is put into it. And the mere fact that some smart people think that it can work is not the reason to abandon it. So very, very cleverly phrased letter. And of course, we got the grant. So that's good. It works. It worked fine. So I continue working on this general area for some years, developed some method that can take what we thought at the time will be a good comparative genomics approach when you look for binding sites. This one was interesting. We didn't follow up with this study, but this was showing that there is a reason that we don't have many avian viruses jumping into humans, and it has to do with the body temperature of the birds. I'm not going to say more on that. And also, we collaborated then with Gary again on when we tried to develop a model for microRNA binding to the mRNA, but combinatorial binding, or not just one microRNA to one mRNA, but a bunch of microRNAs with known protein concentrations to a mRNA. So in that one, we don't use the Boltzmann distribution. We had to use the Fermi-Dirac distribution, also something that we developed together. That was good. Another work that we did at the time was, and it's relevant to what Scott said before, is we figured out that with the exception of the zinc finger proteins, all the other proteins, they have a consensus sequence on which they bind. And then they might modify the surrounding nucleotides. And what we have postulated, I don't know how much it holds. I mean, it looks like it holds. The other proteins, unlike zinc finger, which is only uses the first few amino acids to recognize nucleotides in the major groove, the other proteins, they do bury a whole alpha helix into the major groove. And it's very, very difficult to change amino acids there. So after all, the reviewers at NSF were right, but they didn't know it on that. So here we saw that, yeah, there is this kind of conservation. Practically, you can identify the protein family that binds onto the DNA by looking at the motif of the DNA that they bind. So now I'm going to talk a little bit, I don't know how much time I have, but I'm going to talk a little bit about the something that became very important for me that has not much to do with motives, but following the Gary's advice that you know, have an open mind and follow, you know, what your curiosity point, where the curiosity gets you and everything. What happened was that about 10 years ago, I had a student named Sedgwick with a CS major from Princeton that came to my group and wanted to do some machine learning. So we want to do some machine learning. We wanted to be able to integrate many different data sets and make some inference about the variables in these data sets. So we thought that graphical models might be a good way to go about it. However, there were no these data sets are kind of heterogeneous. They have continuous variables, discrete, and different types of discrete variables. So we needed a framework that can handle with all these variables at the same time. So AJ said, Dr. now Sedgwick, developed such a method where you could learn what's called the causal graph, mainly a directed graph, on this kind of mixed data sets for continuous and discrete variables. And the benefit of that is that you can use this graph either to generate hypotheses, suggest intervention points, biomarker selection, patient stratification, or disease subtyping. Then I had other student, Vinit Raghu, and the postdoc Dimitris Manatakis. They added prior knowledge. They looked on these graphs in the presence of latent confounders. And more recently, Tyler Lovelace, my current student, developed a method where you can run this on tens of thousands of variables, like the whole gene expression data set, by extracting some latent factors about that. So the graphs about that, we have done different publications on that. You can see how you can predict the longitudinal lung function decline and what factors affect it. And you can see the ones that are connected there. You can see what factors might affect culture positivity in ICU patients. More recently, that just published about a month or two ago, where we're looking on a new classification system for new. There is no classification system. There was no classification system. A classification system for a rare disease in children, or finding out what affects the all-cause mortality and the COPD-specific mortality in COPD patients. But what I would like to talk to you is about, let's see how it goes. Okay. It's about, because it's instructive, that's why I want to talk a little bit about it, is this paper that we had a couple of years ago in thorax, which is what we were looking was to see if some variables on the CT scan, together with comorbidities and other clinical variables, can tell us something about whether a nodule we detect in the lung CT scan is cancerous or not. The reason for that is that there is the CT scans are much more sensitive in picking up these nodules than the X-rays. However, they're 96%, they have 96% false positives. And that creates a problem because you cannot go back to the patient and say, look, we found something, but I'm 96% sure that you are okay, don't worry. You need to follow up that every 3 months or 6 months, do follow-up CT scans. So you do follow-up CT scans, unnecessary biopsies. You increase the anxiety because even if they tell you, I'm 96% sure you are okay, you are still not going to be sleeping very well at night, and increase the healthcare cost. So what we looked, we put the variables that we had in this graphical model, and we found, these ones are the CT scan variables. This is the comorbidities, and these are the demographics. And as you see, they can be all in the same model. And we identify 3 variables that you will see they give pretty good predictability. But the reason that I mentioned that is because these models can also be used to help us identify biases in the data sets that we have. For example, here, the emphysema and bronchitis are not linked to the cancer status, although we know these are risk factors for lung cancer. So why they are not linked? So we had to go back to the data, look at the data, and what happens is that this was a clinical cohort, and in order to get into the cohort, you need to be a heavy smoker and not have any prior symptoms or anything like that. So that means that both the control group and the cancer group, they had similar bronchitis and emphysema percentages. Therefore, that gives the limitations of the model. Now you say, this is not for the general population. This is only for heavy smokers that they have X number of pack years, right, on that. And that helps you when you would like to apply the model. You cannot do that with the deep learning, or at least not so easy, with deep learning and other methods. Of course, this model was better than other models. Of course, I mean, that's not a cool thing. You can work with a model long enough, you can make it better than other models, but that was the cool thing. So with the other model, the classical, the Brock model, you can see that there is no clear separation between those that they have cancer and those that they don't have cancer, right? But in our model, you can threshold it here, and anyone below that, it's clear. It doesn't have cancer. And this is 28% of the 96%, which were the false positives. So if this is validated in a larger cohort, that will be a very big big thing on saving some people with further procedures. And that's why my my clinical collaborator gave some NPR interview and so on. So that's all I wanted to say. So I forgot to mention the other thing is that when I joined Gary's lab, he impressed me that I'm an early riser. I was going to the office at 7:30, between 7:30 and 8, and he was there already. And then he was leaving after 5 o'clock. And after a while, you feel very bad to go to your work after your boss and leave before the boss. So that's the lead by example, you know, on that. The other one I mentioned was non-judgmental in when I wanted to go to Celera, and highly collaborative. And just to tell you the story very briefly about the band that I said before. So Gary told me once that when he graduated from the bachelor's degree, he took a year off and went with his band to tour the United States. I don't know, they like that. And during that time, it was the time where a lot of sequences started depositing into the public domain. So when he came back, there was these sequences and say, oh, that's something that I would like to do on that. And that's how he started his PhD with Larry. So Gary, thank you very much for all the things that you offered us and me personally. Congratulations on your retirement, and thanks everybody for your attention.